On this week's GARNet Research Roundup, I'm really delighted to be joined by Professor John Christie from the University of Glasgow. And we're going to be talking about a paper just published in Plant Physiology, the title of which is De-etiolation Enhances Phototropism by Modulating Non-PHOTOTROPIC HYPOCOTYL 3 Phosphorylation Status. Okay, a bit of a mouthful of a title there. But thanks so much, John, for joining us today. It'd be great if you give us a brief overview about this paper, please.
Sure, and good to talk to you, Gerd. So the focus of the paper was essentially to get a better idea of the mechanisms that underpin phototropic responsiveness in plants. So phototropism is the directional growth response of plants towards or away from a light source. In stems, the growth is towards the light. This is a response that plants adhere to when they emerge from the soil. So it's really important for seedling establishment. So plants have to sense the direction of light in order to kind of reprogram their development. So they undergo photoautotrophic growth under light conditions. Mm-hmm. So in the dark, they're primarily undergoing heterotropic growth. So this transition is really important as they emerge from the soil.
Yep.
The process phototropism is a process that we've studied for many years. So typically, you do this artificially in the lab. Mm-hmm. You study the growth of seedlings towards a directional blue light source.
Yep.
And typically you use etiolated or dark-grown seedlings.
Mm-hmm.
A few years back, we started to transition this and use light-grown seedlings instead of dark-grown seedlings. Yep. And the basis of this paper essentially shows that the response is different when you're using light-grown seedlings. In light-grown seedlings, the response appears to be somewhat faster. There's a faster onset of the response towards curvature, towards the light. And what we think is happening there is that once you've reprogrammed your development for photoautotrophic growth, then it's really more important that you establish yourself and capture as much light as possible for photosynthesis.
Okay.
So when you're in the dark, light can also be dangerous. If there's too much light there, it can be phototoxic. So what we have, I think, in the dark is some sort of brake mechanism in place whereby you don't overcompensate growing towards the light when conditions are more sensitive.
Okay, so that's a key distinction for those of us who aren't, real experts on light and plants. It's the distinction between kind of generalized light signal versus a directional light signal, and that's what you're showing here. You found in this paper that the de-etiolation, so the greening response, is independent of light color.
That's correct. So the receptor systems that we work with that control the phototropic response, these are called the phototropins. Mm-hmm. So these are receptor systems that don't elicit de-etiolation, so the photomorphogenesis of plants as they transition from dark-grown development to light-grown development. Mm-hmm. Okay. So these are primarily mediated by other receptor systems. So these are the cryptochromes, which are also blue light receptors, as well as the phytochromes, which are red light receptors. And wavelengths, red and blue light, can promote de-etiolation or photomorphogenesis of higher plants.
Mm-hmm. Right.
So de-etiolation by either phytochrome or cryptochrome is sufficient to prime the faster phototropic response that we see in these seedlings.
Okay. So part of the paper, the first part of the paper, really is about defining this response. But then there's a lot of information about the molecular players about this and what you know about this. So the title talks about this NPH3 protein. So can you give us a little background about what that does and how that is affected by this light signal?
Sure. So NPH3 was a protein or a gene that was identified by Manny Liscombe's group 2 decades ago now. Right. And this is a protein that appears to be instrumental for coordinating hormone fluxes that drive this differential growth of the seedling to grow towards the light. The function of this protein, non-phototropic HYPOCOTYL 3, or NPH3, is very poorly understood. Mm-hmm. And so we really don't have a handle what its biochemical function is, but it seems to be communicating directly with the receptors that detect the direction of the blue light, and the protein seems to be directing transport or accumulation of the plant growth hormone auxin on the shaded side of the stem to promote cell elongation, and this differential cell elongation across the stem is what drives that curvature response. Right. But studies today have been it's been very challenging to actually dissect what this molecular player is actually doing.
But as part of this paper, you show that the phosphorylation status is changed in these different treatments, so the de-etiolated and the ethylated.
Also, when the NPH3 protein is activated, if you will, by the photoreceptor under blue light, one readout that you can measure is that this protein becomes dephosphorylated in response to light treatment. So in the dark, this molecular player NPH3 is in a phosphorylated status, and then in the light it becomes dephosphorylated. The function of this is not fully understood, but what we find from these studies is that this level of dephosphorylation seems to be affected when you have seedlings that have been exposed to photomorphogenesis. So light-grown seedlings have a different balance of dephosphorylated forms of NPH3 compared to phosphorylated forms of NPH3. So what we propose in the paper, given the results that we get, is that it appears that the phosphorylated form of NPH3 seems to be a big player in instigating phototropic responsiveness, whereas when you have a dephosphorylated form of the player, this is not active in terms of bringing about the response.
And the details of that, no doubt, will hopefully come out in the future, what exactly that phosphorylation is doing, indeed.
Exactly, yes. Exactly. This will be a challenge. So identifying the sites that become dephosphorylated, sites that become phosphorylated, the molecular players that control this phosphorylation, dephosphorylation, will be a big challenge in terms of dissecting. Yeah,
so as part, some of the experiments you use a phosphatase inhibitor, and you show that alters the movement of this, but you don't have any idea about the actual phosphatases that are acting on NPH3 at the moment.
No, not as such. So we've done some experiments looking at kinases and phosphatases that will interact with the NPH3 protein. So we do have a kind of molecular handle and potential players, and this is something that we plan to pursue in the future.
So actually, what would be happening to a seed or a newly germinating plant in the soil? What exactly do you think is happening in the real world in this example?
I think here, so there's two different conditions happening. So there's emergence of the seedling from the soil, so underneath the soil as it comes out and is exposed to the light environment. If it's emerging under sort of a canopy in a shaded environment, then it really uses that directional information to direct its growth towards the light source in order to change its development into photoautotrophic growth. For established seedlings, the situation is going to be different. So for an established seedling, they've already transitioned into photoautotrophic growth, and there'll be the surrounding light environment might be more diverse, so it might be fluctuating quite a lot. So if it can respond to changes in more detail in terms of fluctuating changes, then this might be a competitive advantage of having a faster phototropic response in light-grown seedlings.
Yeah, I see. You've also, congratulations, you've had a science paper out recently with Mike Blatt, and as part of that paper, a key concept was about this fluctuating light environment as well, right? So I think the experiments that have been done in constant light, that's not a real life situation. So having this movement and dark shade and light is the a true test of what's going on in the environment.
Yeah, I think so. When we think about trying to manipulate these types of responses, you can view evolution. Probably the conditions, if you think about the light environment, it's highly flexible, it's variable. So when you're trying to engineer, in this paper in Science, if you're thinking about mechanisms that limit photosynthesis, then nature will have evolved things to still be flexible, but not possibly optimal. So there's scope for improvement there in terms of synthetic engineering, and that's something that we pursued within the science paper.
Cool. All right. Well, thanks so much, John, for joining us today. It's been great to chat about this paper. Thanks.
Pleasure. Thank you.